Hello, my name is Patrick Adelberger, and I'm a PhD student at Johannes Kepler University in Linz, Austria. Today I'm going to talk about Core, a web-based visual analysis tool for creating and characterizing cohorts. Core is a joint work between the Visual Data Science Lab at Johannes Kepler University and Boehringer Ingelheim. There are some tools that allow users to create and analyze cohorts. For example, cBioPortal. But cBioPortal has some limitations. It is hard to define cohorts based on mutation data. It's also hard to define cohorts based on the absence of one or more multiple mutations. It also doesn't support the iterative process of defining a large set of cohorts. For example, creating cohorts by multiple subsequent split operations. And you cannot keep track of the cohorts that you generated. There are also other tools out there that allows the user to create cohorts. One of those is Composer by Rogers and others, as well as Kava by Sheng and others, and the prostate cancer analysis tool by Bernard and others. All those tools can create cohorts, but they do not allow the creation of multiple cohorts at once. And it's also not easy to keep track within those tools and also see the relation between them. In those tools, it's also not possible to visualize the data of multiple cohorts at once. That's why we developed Core, to characterize and create multiple cohorts and track the relationships between those cohorts. So if you load the Core web application, you come to a landing page where we have additional information about Core, which datasets it uses, which features it has, all the publications related to it, and also some onboarding steps to help new users. Here's a QR code to the application as well as a link to it. The publicly deployed Core instance contains 5 datasets. The database contains mutation data from the AACR Project GENIE, mRNA expression data, copy number, DNA copy number, and mutation data from the Cancer Genome Atlas, and data from the Cancer Cell Line Encyclopedia, as well as 2 depletion screen datasets. In total, Core's database contains data of over 128,000 samples. The pre-processing steps are described in the paper's supplementary notes. To demonstrate Core, we use a use case that is based on the publication by Nazar in the New England Journal of Medicine. Here the authors focused on the GENIE patients and assess race and gender differences with respect to the KRAS G12C mutation frequencies in non-small cell lung cancer and colorectal cancer patients. The KRAS gene is mutated in around 15% of all human cancers, making one of the most frequent mutation cancer-causing genes. The G12C is one of the most prevalent KRAS mutations. So if you load the application, you come to the landing page as already mentioned. On the top right corner, you see the Start Analysis button. If you click on it, you will get to the analysis tool. You have here some additional information that would help you throughout the process. The text within the blue box is some onboarding steps that you can follow. We select the GENIE dataset, which then is represented in the rectangle, with the size at the bottom and also the name of it. If you select now this cohort, you can see it is added to the bottom, the action view, where we work with the selected cohorts. We split it Core into 2 areas. The top area is the cohort evolution view, where we keep track of the cohort, as you will see later on in the use case, and the bottom half, where we work with the selected cohorts. We also highlight the selected cohorts with different colors to keep a better track of them. So there are different operations you can do to the selected cohorts. You can either characterize them or create new cohorts. The first operation is the view operation. It is the main route to exploring the dataset. It allows to visualize an attribute of a cohort and analyze or compare the different attributes of cohorts. The filter and split operation is based on the view operation, where you can interactively create new cohorts. Then we have the prevalence operation, which is a dedicated visualization to explore the prevalence of the selected cohorts. It is also possible to analyze the individual items in each cohort to see if there are outliers or not within it. And the last operation is the compare operation, where it's possible to find out if there is a statistical difference between cohorts based on one attribute. So let's start with the view operation. Depending on which attribute you type in, you get different visualizations. For example, if you use age, a quantitative attribute, we'll see a density plot. If we select gender, a categorical attribute, we see a bar chart. We can also combine different attributes. If we add now age to gender, we get a box plot. Different combinations of attributes result in different visualizations. If we would combine two categorical attributes, we would get an area chart. If we combine two quantitative attributes, a scatter plot. Now let's start with the first cohort for the use case. What we need now is to create a cohort that includes the major subtypes for the non-small cell lung cancer cohort. Those tumor types are the lung adenocarcinoma, the lung squamous cell carcinoma, and the non-small cell lung cancer. If we click now on filter, we will generate a new cohort that includes all the values that we selected in it. If we now add the cohorts to the cohort graph, we then see that it's also automatically selected, and we also see that the size difference to the dataset in the third row of the cohort representation. We now create another cohort, the cohort for the colorectal cancers. Here again we select subtypes that are contained within the colorectal cancer, which are colon adenocarcinoma, colorectal adenocarcinoma, and rectum adenocarcinoma. Again, if we use the filter operation, we will create a new cohort that includes all those values within one cohort. To get an initial impression of the demographics, we select both the cohorts now and look at the age distribution between them. As you can see, the patients in the colon cancer cohorts are on average older than the ones in the lung cancer. To confirm that difference, we will now use the comparison operation. We go back to the operations and select compare. Here we use the visualization technique Tordino by Eckelt and others. It creates a matrix of pairwise comparison based on the attributes selected. Here, age. You can see additional information about the test and that the difference is statistically relevant. We can also analyze the gender between those two cohorts. Again, if we click on the cell, we get additional information about the tests used and also a visualization that shows the difference of the attribute values. So now let's create the different cohorts for different races. Here we will focus only on the lung adeno or non-small cell lung cancer patients. So we only select this one cohort. And then we choose race. And we will select the top 3 most prevalent categories for race. And if we click on split, it will generate 3 cohorts with for each category we selected. The 3 created cohorts are Asian, Black, and White. We now automatically select all the race cohorts and split them by gender. Per default, all categories are selected, so clicking on split will automatically create a cohort for each category. Now we generate this 6 cohorts based on the gender. We will now have a look at the KRAS amino acid mutation. And as we can see here, that the KRAS G12 mutations in the non-small cell lung cancer are less frequent in the Asian women compared to Asian men, so the blue and the red, but are more frequent in Black and white women compared to Black and white men. These observations match the findings by Nassar and others. These findings were easy and fast to reproduce with Coro, which allows the user to create and analyze multiple cohorts at once. Now let's select all the different categories for those G12C mutation and create a new cohort based on them. We'll now use those 6 newly created cohorts to have a closer look at the prevalence of those cohorts. So we go back to the operations and select the prevalence operation. Here for each selected cohort we have a bar chart. This long bar, this light gray bar represents the dataset. The dark gray bar, the reference cohort, and the colored bar, the selected cohort. In this case, the colored bars or the selected cohorts are so small compared to the dataset that they are not visible. Above the bar, you have multiple checkboxes. The one that's grayed out represent the filter steps taken to create the selected cohort. You can use those filter steps to then create reference cohorts. So that means, for example, if I now select the filter steps gender, race, and tumor type, I will create a reference cohort that includes all non-small cell lung cancer patients that are Asian and female and compares them to our initial or the selected cohort, which is also filtered by the KRAS 12C mutation. Because it's so hard to see of the small sizes of these 2 cohorts compared to the dataset, you can also hide the dataset cohort. Then you have a better overview between the reference cohort and the selected cohort. We can also analyze the cohorts on the single data items. We can here add age and gender as attributes of interest, and then we will get a table which uses the lineup technique by Kratzel and others. Here you can then also see a dedicated column to represent the cohorts the data item is in. It is also possible to export the current table as a CSV file. If you are finished with the analysis session, you can also share the session with somebody else. For that, you just save your session, give it a name. You can also make it private, so it's only for you, or you can make it public and then save the session. You now will get a new link or URL that you can share with somebody else. And I'll just demonstrate it if opening it in a new tab where I copy in the link from before. And as you can see, it automatically creates the same cohorts that I had before. It goes step by step to create them. The only difference now is that the cohorts are not selected, but that you can do anyways manually. For future work of CORL, we thought about supporting longitudinal data, which is currently not supported. Longitudinal data is data from multiple samples of one patient at different time points or detailed treatment history. We're also thinking about adding an automatic compare operation that allows the user to select cohorts of interest, and CORL then does the calculation to find the attributes that differ the most between those cohorts. Technically and conceptually, it's also possible to use CORL for other data and fields. For example, analyzing the data of COVID-19 patients of a country. Therefore, we also want to support the upload of custom data. So in this talk, I quickly showed how easy it was to reproduce the use case of the mentioned paper. It also showed how to interactively create multiple cohorts and how to analyze them with the visualizations. So try it out yourself and give us feedback about what data sets you think could be also beneficial to include or other features. This concludes my talk, and thank you for watching, and I'm open for any questions.